Welcome back to the deep dive, where we really get into the nitty-gritty of new research and pull out the key stuff for you.
Today, we're looking at a real evolutionary head-scratcher. It goes back, way back, like 500 million years. We're talking about the very first plants to colonize land, the bryophytes.
Right, so that's mosses, liverworts, hornworts. You see them everywhere.
Exactly. And they are the oldest lineage of land plants still around.
Which is kind of amazing when you think about how simple they look, right? They don't have that complex internal plumbing, the vascular system that, say, a flower or a big tree has.
No, structurally, very basic. But they are absolute survival experts. You find them thriving in places you wouldn't expect anything to grow. Volcanic ash, Arctic ice fields.
They've been doing this since before the dinosaurs even showed up.
Which leads us to the big paradox. How have these, well, simple organisms managed to stick around for 500 million years, outlasting so many other life forms?
When they actually have fewer genes, on average, than most modern plants, that seems backwards.
It really does. You'd instinctively think, more genes equals more complexity, more resilience.
Yeah, wouldn't fewer genes suggest they just kind of stalled, evolutionarily speaking? Why aren't they just relics?
See, that's the core question this huge new study tackles. If they were stalled, they wouldn't be flourishing globally. The answer, it turns out, from this big super pangenome analysis isn't about the sheer number of genes.
Right.
It's about the sheer variety in their genetic toolkit, diversity over quantity.
Okay. So our mission for this deep dive is to unpack that. We're digging into the results from sequencing, 123 new bryophyte genomes?
That's right. And we're going to show you how these ancient plants built this system that favors constant innovation over just copying the same old genes.
So let's set the stage first. Remind us about the basic plant family tree split.
Right. So around 500 million years ago, land plants split. Two major groups emerged that still exist today. The bryophytes we're talking about, and the vascular plants, or tracheophytes, basically everything else. Trees, ferns, flowers.
And the key difference isn't just size or those internal pipes, it's their life cycle you mentioned.
Exactly. This is crucial. Bryophytes, the moss you see on a rock. That visible green part is mostly the gametophyte generation. It's typically haploid, one set of chromosomes.
Okay.
Vascular plants, like that oak tree, they're dominated by the sporophyte generation. That's the big complex structure. And it's diploid, two sets of chromosomes. Keep that haploid-diploid difference in mind. It's central to their whole genetic strategy.
Got it. Haploid gametophyte for bryophytes, diploid sporophyte for vascular plants. Okay, let's talk about the study's scope. This wasn't just a few random moss samples.
No, far from it. This was massive. They sequenced 123 new high-quality genomes, and they really aimed for coverage representing 47 out of the 55 known bryophyte orders.
Wow.
Yeah. 37 liverworts, 82 mosses, 4 hornworts, really filling in a huge gap in our understanding of plant genomics.
And this massive data set confirmed that initial point bryophytes do have fewer genes overall.
Yep. On average, just under 28,000 genes per bryophyte genome. Compare that to vascular plants, averaging nearly 35,000.
So if it's not the total count that matters for their success, how do we measure their genetic potential?
You shift focus. Instead of just counting every single gene, you look at the number of unique types of genes, or gene families. The study calls them orthogroups.
Orthogroups? Like job roles in a factory. Instead of counting all employees, you count how many different jobs there are.
That's a great analogy, exactly. Are you just duplicating the same assembly line workers over and over, or are you adding unique specialists, designers, engineers? We're measuring the variety of functions.
Okay, that makes sense. So they analyze, over 1 million orthogroups across hundreds of species?
Yes, across 343 species in total, including algae for comparison. And they broke these orthogroups, these gene families, into 3 key categories. Understanding these is really the core of it all.
All right, let's break them down. What's the first category?
First up, core gene families. These are the absolute essentials, found in 80% or more of the species within a lineage. Think basic metabolism, photosynthesis, cell structure.
Yeah.
The foundational stuff every plant needs.
The standard operating procedures for being a plant?
Pretty much. And interestingly, vascular plants actually have a few more of these core families, about 6,600 compared to roughly 6,200 in bryophytes. So slightly more standardized parts.
Okay, category 2.
Accessory gene families. These are more specialized. They're present in at least 2 species, but in fewer than 80%. So not universal, but shared among some members.
Like optional extras, rust-proofing maybe, or a special cold weather package.
Perfect analogy. Genes for dealing with specific stresses, unique environments, particular defenses. Only certain lineages or species adapted to those niches would have them.
And the third category?
Unique gene families, sometimes called orphan genes. These are found in only one single species or taxon.
Ah, so these are the truly novel inventions, the brand new prototypes that only one specific factory line has developed.
Exactly. They represent very recent or lineage-specific innovation. True genetic novelty.
Okay, core, accessory, unique. So if the bryophyte story is about diversity and resilience, we'd expect them to really shine in those last two categories, right? Accessory and unique.
And that is precisely what the data shows. It's quite dramatic, actually.
Lay the numbers on us.
When you tally up the total number of distinct gene families, the non-redundant count bryophytes have way more, nearly 638,000 compared to about 374,000 in vascular plants.
Wow, that's a huge difference. Where does it come from?
Almost entirely from those novelty categories. Bryophytes has significantly more accessory families, over 4,000 versus less than 1,600 for vasculars. And they average way more unique families per species, almost 3,900 compared to just over 2,200.
So let me get this straight. If you pick up a random moss, a huge chunk of its genetic identity is made up of these specialized or totally unique gene types.
A massive chunk. Those accessory and unique families together make up 56% of the gene families in a typical bryophyte genome.
56%, compared to what in vascular plants?
Only 36%. It highlights a fundamentally different evolutionary strategy.
Okay, spell that out. What are the two strategies?
Vascular plants tend to expand their capabilities mainly by duplicating their existing core genes. More copies of the reliable stuff makes them robust, often bigger and more complex structurally. Bryocytes, on the other hand.
They prioritize breadth, having a wider range of different tools even if they have fewer tools overall.
Precisely. They focus on genetic diversity, constantly adding novel functions. Left total DNA, maybe, but packed with unique innovations. It's maximizing potential while keeping the genome relatively streamlined.
Okay, that's a major insight. So the next big question is how? How are they generating this constant stream of new genetic material? Where does this novelty come from? Let's tackle the first mechanism, de novo gene origination.
Right. De novo meaning from scratch. This is one of the most fascinating ideas in genomics, I think. It's the idea that functional genes can literally arise from DNA sequences that previously didn't code for anything.
What people used to call junk DNA.
Exactly. Though we know now it's often far from junk. This process seems to be a major engine driving that huge number of unique and accessory genes we just talked about in bryophytes.
But how can scientists be sure a gene is brand new, truly de novo, and not just some really old gene that's mutated so much it looks different, or a copy that got lost in other species?
Good question. There are telltale signs, kind of like genetic fingerprints of youth. These newly formed genes, the ones in the unique and accessory categories, often have a simpler structure.
Simpler how?
They tend to have fewer introns, those non-coding bits that interrupt the coding sequence in many eukaryotic genes. And their overall coding regions are often shorter compared to the ancient core genes. This matches what we see in young genes identified in other organisms, like rice or Arabidopsis.
And crucially, they're not just modified copies of existing genes.
The evidence strongly suggests not, for a large portion of them. In well-studied bryophytes like the moss Physcomitrium patens, they found that around 85% of the unique genes showed no significant sequence similarity to any known orthogroups.
85%. So they really did just appear, sprung from non-coding DNA.
That's the strong implication. They didn't come from duplicating and modifying something that was already there.
Yeah.
They originated directly from sequences that weren't genes before.
Wow. Okay, can we trace how that happens? Did the study give any clues about the actual process of a non-gene becoming a gene?
Yes, they did some really neat work looking at the liverwort Marchantia polymorpha. They compared closely related subspecies that diverged relatively recently, maybe 5 to 7 million years ago.
And what did they find?
They found that a large percentage, maybe 70–80%, of the orphan genes, those unique ones in one subspecies, lined up perfectly with sequences that were clearly non-coding in the other closely related subspecies.
Ah, so they caught them in the act, almost. You can see the non-coding precursor in one and the newly minted gene in the other.
Sort of, yeah. It points to these non-coding regions acting as protogenes, raw material waiting for the right mutations.
And what kind of mutations turn a protogene into a real gene?
The key seems to be messing with stop codons. A stop codon is a signal in the DNA that tells the cellular machinery to stop translating the sequence into protein. Non-coding regions are usually littered with them, so if they get accidentally translated, you only get tiny, useless fragments.
Right.
But mutations can gradually eliminate or change these stop codons. As that happens, the potential reading frame gets longer and longer until eventually it might produce a stable protein sequence long enough to potentially do something useful.
So evolution is essentially cleaning up the stop signals, allowing a functional protein to emerge from the noise.
That's the model, and they saw evidence for it. The slightly older young genes tended to have fewer stop codons than the very newest ones, showing that progressive elimination.
Okay, that's amazing. De novo origin is powerful, but why is it apparently so efficient in bryophytes? You mentioned their life cycle earlier.
Yes, this brings us back to the haploid advantage. Remember, the main visible photosynthesizing body of a moss or liverwort is gametophyte, meaning it's haploid. It has only one set of chromosomes.
Unlike us, or an oak tree, which are diploid two sets, how does being haploid help?
Think about it. If a brand new gene pops up via de novo origin in a haploid organism, there's no second copy of that chromosome to potentially mask its effect.
Ah, so the new gene's function, good or bad, is immediately expressed. It's instantly exposed to natural selection.
Exactly. There's no hiding place. It's like instant beta testing by evolution.
Right. If it's harmful, that individual plant likely doesn't survive or reproduce well. Zapped.
But if it's beneficial, maybe it helps tolerate drought a tiny bit better or resist a local fungus, that advantage is immediately felt, and because it's beneficial, it's much more likely to be passed on and spread through the population quickly.
So the haploid state speeds up the fixation of useful new genes.
Massively. Plus, many bryophyte tissues have two-tipidency, meaning almost any cell can potentially regenerate into a whole new plant. This also helps propagate beneficial mutations rapidly.
Okay, so they have this incredibly efficient internal engine for creating brand new genes from scratch, turbocharged by their haploid life cycle. That explains a lot of the novelty. But you said there were two mechanisms.
That's right. The other major source of novelty isn't internal creation, it's external acquisition. Mechanism 2 is horizontal gene transfer, or HTT.
HTT borrowing genes sideways from completely different organisms, not inheriting them vertically from parents.
Precisely. We're talking about picking up genes from bacteria, from fungi, viruses, maybe even animals, though that's rarer. And the study shows that for bryophytes, this isn't just a rare occasional event like it seems to be in many other complex organisms.
It's more common for them.
It seems to be a continuous, ongoing process, a steady stream of genetic innovation flowing in from the wider biological world.
Do the numbers back that up? More HTT genes in bryophytes?
They do. On average, the bryophyte genomes they analyzed contain more HTT-derived genes, about 229 compared to tracheophytes, which averaged around 163.
And how do these foreign genes even get integrated? Does the bryophyte genome just accept them easily?
There seems to be a mechanism facilitating it. These HTT genes are often found nestled within or near regions of the genome that are rich in long-terminal repeats, or LTRs.
LTRs, those are often associated with mobile genetic elements like retrotransposons, right?
Exactly. The idea is that these LTR elements might be helping to integrate the foreign DNA, or perhaps helping it to proliferate once it's inside the bryophyte genome. They might act like genomic landing pads or facilitators.
Okay, so they're constantly acquiring foreign genes, and their genomes might even help slot them in. But what are these genes doing for the bryophytes? Are they useful?
Oh, absolutely crucial, especially for dealing with stress and defense. It's like they're outsourcing some of their toughest survival challenges.
What kind of stress?
Abiotic stress, heat, drought, UV radiation. In the Moss Fiscometrium patents, they tested the expression of known HTT genes under stress conditions. A staggering 93% of them showed changes in expression.
93%. So they're clearly involved in the stress response network.
Deeply involved. Take the HPA3 gene. Moss has picked this up from bacteria. It encodes an enzyme that detoxifies harmful compounds, specifically D-amino acids, that can build up when the plant is stressed or starving.
So a bacterial gene helps the moss stay stable in really harsh, nutrient-poor environments.
That's the function, yes.
Yeah.
Essential for colonizing those tough habitats where they thrive.
Okay, that covers environmental stress. What about defense? Fighting off pests or competitors?
Even more striking examples there, often involving genes picked up from fungi. Liverworts, for example, acquired a fungal gene called YANB. It gives them direct antibiotic activity against other fungi and bacteria competing for the same space.
Chemical warfare using borrowed weapons.
Pretty much. Another fungal acquisition in liverworts is the EUPF gene. This helps them synthesize a complex molecule called upenifeldin. And this stuff is potent. It has broad-spectrum antifungal, antimalarial, and even anti-worm properties.
Wow. So they're packing some serious multi-purpose chemical defenses courtesy of fungi.
They really are.
Yeah.
But perhaps the most stunning example, the real showstopper in the study, relates to insect defense.
Go on.
There's a gene called FBT, also fungal in origin, found in many liverworts and mosses. It codes for a protein containing what's called an FD-lectin domain. Lectins can often interfere with digestion in insects.
Okay, so it sounds like an insecticide gene.
Uh,
Did they test it?
They did. They isolated this FBT protein from a liverwort species, Acrolegunius anvicensis, and they fed it to larvae of the cotton bollworm, Helicoverpa armigera, a major agricultural pest.
What happened to the bollworms?
The results were dramatic. At a really tiny concentration, just 0.45 micrograms per milligram of diet, this liverwort protein caused 97.62% mortality in the larvae.
Oh, 97.6% from a gene this little plant picked up from a fungus millions of years ago.
Exactly. That level of potency is right up there with, maybe even better than, some commercial bioinsecticides like butoxin that we rely on heavily in agriculture. It also badly stunted the growth of another pest, the fall armyworm.
That is absolutely incredible. It really hammers home the power of HGT for these organisms. They're not just surviving. They're acquiring cutting-edge biochemical tools from across the tree of life.
And that really ties everything together. The long-term resilience, the ecological success of bryophytes, it's driven by this dual strategy. Constant internal innovation through de novo gene birth, supercharged by their haploid state.
And continuous external acquisition of optimized solutions, particularly for stress and defense, through HGT.
Right. And the net result is that remarkable genome structure, where over half, 56% of their gene families, fall into those specialized accessory and unique categories. It's a toolkit built for adaptability.
So what's here? The big takeaway is that this unique genetic approach directly explains their incredible staying power and ecological range. They've colonized almost every environment imaginable using this diverse set of tools, including novel immune receptors and those diverse secondary metabolites, many of which likely came from HGT or de novo events.
It really challenges our typical view of evolutionary success, which is often associated with the structural complexity and gene duplication seen in vascular plants.
Yeah, it presents two very different paths. Vascular plants, robustness through duplication, maybe fewer types of tools, but lots of copies. Bryophytes, hyper-efficiency in generating and absorbing novelty, keeping the toolkit diverse and adaptable.
Exactly. One path emphasizes reliability and scale. The other emphasizes continuous innovation and flexibility.
So we'll leave you, the listener, with a final thought to chew on. Which strategy ultimately proves more resilient over the vast stretches of evolutionary time, building robustness through duplication, or constantly weaving in novelty through de novo creation and horizontal transfer?
And perhaps more immediately relevant.
Yeah.
Given the power of some of these HGT genes we've seen, like that potent insecticide from the FBT gene, what other untapped potential lies waiting in the genomes of these ancient unassuming plants? What solutions for agriculture, medicine, or biotechnology might be hiding in plain sight on a damp rock?
Definitely something to think about. The potential implications be huge.
A vast, ancient library of genetic solutions we're only just beginning to read.
Thank you for joining us for this fascinating deep dive into the genetic secrets of the bryophytes.